Hello everyone and welcome to the new session of the lecture series, and today's topic is cell cycle and cell division. And today we'll be discussing about all the questions from the NCERT book. So this lecture is entirely NCERT based. Now coming to the first question. So the first question is, what is the average cell cycle span, means the duration of the cell cycle. Here they are asking about the duration. Okay, what is the duration of the cell cycle of a mammalian cell? So the answer is, typically the duration of the cell cycle of a mammalian cell is roughly around, or you can say approximately 24 hours. Alright, so this was simple. Next question is, distinguish between cytokinesis and karyokinesis. So we already know that what is cytokinesis. Cytokinesis is division of cytoplasm, whereas karyokinesis is division of nucleus, or we can also say nuclear division. Alright, so what is the difference between cytokinesis and karyokinesis? Alright, so let's see some of the difference between the nuclear division and the cytoplasmic division. So how can we define karyokinesis? Karyokinesis is simply a biological process involving nuclear division. Alright, and karyokinesis is further divided into prophase, metaphase, anaphase and telophase. Okay, depending on the condition of the chromosome. Whereas what is cytokinesis? Cytokinesis is a biological process which involves the cytoplasmic division, and it is not further subdivided into some other stages. Now what happens during karyokinesis? The nucleus gets divided into two daughter cells. Alright, so if this is the nucleus of the cell, it gets divided into two cells or two nucleus. Alright, whereas in cytokinesis what happens? The cytoplasm, the organelles, the nuclei, everything, they are undergoing division and are equally distributed to the daughter cells. Okay, so this was the differences between the karyokinesis and cytokinesis. Now let's come to the third question, and this is a very important question. Alright, so here we have to describe the events that take place during the interphase. Alright, interphase we already know is a resting phase. Okay, interphase is a resting phase where the cell is preparing itself for the cell division. Interphase is further divided into G1 phase, S phase and G2 phase. Alright, now let's talk about the events that take place during the interphase. Now coming to the G1 phase, which is also known as the gap 1 phase. Okay, so what happens here? Cell growth is taking place and the cell is metabolically active. We say that interphase is a resting period, but actually the cell is metabolically active and it is producing lot of carbohydrate, lipids, RNA as well as proteins during the G1 phase. Okay, what else is happening during G1 phase? There is no DNA replication, means there is no change in the DNA content during the G1 phase. Now let's talk about the next phase, which is the S phase or the synthetic phase. In synthetic phase what happens? DNA synthesis, or you can say DNA replication is taking place. Okay, some proteins known as histones are synthesized Okay, and what is happening? Due to DNA replication, the DNA content per cell is double now. Okay, the DNA content doubles. Okay, if initially the DNA content was say 2C, okay, in any diploid organism means 2N, then the DNA content after the S phase will be 4C. Okay. Now what else is happening during the synthetic phase? DNA doubles, but there is no increase in the chromosome number. Chromosome number remains the same. Means if the organism was diploid, say 2N, it will again after the S phase contain 2N number of chromosome, means 2 sets of chromosome. But the DNA content will be 4C. Here it was 2C, but the DNA content after S phase will be 4C. Another thing happening during the S phase is that in some animal cells, the centrioles will duplicate. Okay, they self-duplicate. And what is the role of centrioles? Centrioles help in the assembly and the organization of the microtubules. And microtubules later form the spindle fibers. Now let's talk about the third phase known as the G2 phase, or you can say gap 2 phase. Here what happens? Cell growth is taking place. Okay. Here also cell growth is taking place and the cell is preparing itself for the cell division. So we say final preparation for the cell division is taking place during the G2 phase of the interphase. Now what happens during G2 phase? Cytoplasmic organelles such as the Golgi apparatus, or you can say Golgi complex, mitochondria, okay, you can write ER also, endoplasmic reticulum. These divide and they double, okay, and then they are separated later inside during cytokinesis to each cell, okay. What else is happening during the G2 phase? Synthesis of some protein known as tubulin protein is taking place. And what happens? Tubulin later helps in the formation of the spindle apparatus. Alright. Along with these tubulin proteins, some other proteins are also produced during the G2 phase. Okay, you can write one example. Some cyclins are produced during the G2 phase of this cell cycle. And what is the role of cell cyclins? They help to regulate the cell cycle. They help to regulate the cell cycle. Alright, so this was about the events that take place during the different phase of interphase. Now let's talk about the another question, which is what is the G0, also known as the quiescent phase of the cell cycle. Alright, so let's talk about the G0 phase now. Now interphase, you know, is divided into G1 phase, S phase and G2 phase. Okay, sometime what happens? Some of the cells such as cardiac cell, you can say heart cell, skeletal muscle cells, some lymphocyte and some nerve cell. Okay, they stop dividing and what they do? They enter the G0 phase. Okay, so what is happening here? This is the G1 phase. So what do the cells do? What these cells do? They stop dividing and they exit the G1 phase and they enter the G0 phase, okay, which is known as the inactive phase. This is the inactive phase, okay, where the cell is metabolically active, okay. The cell is metabolically active, but the cell is not dividing. It no longer proliferates, okay. But what happens when the conditions change, okay? And depending on the requirement of the organism, the growth in this cell can resume, okay? And what happens then? Then these cells can again re-enter the G1 phase, all right? And then the cycle will continue, okay? And then we have the M phase, mitotic phase, all right? So you can say some cells like these cells, heart cells, skeletal muscle cell, lymphocyte and nerve cells, they do not divide further and exit the G1 phase and enter an inactive stage called the quiescent stage or the G0 stage of the cell cycle. Alright, and depending on the conditions, depending on the requirement of the cell, these cells can resume their growth and enter the G1 phase again. Now let's talk about the fifth question of the NCERT book. Why is mitosis called the equational division? Okay, we know in mitosis what happens. If the cell is diploid, after mitosis, it will still remain diploid. Okay, means it will contain the same number of chromosomes as the parent cell. Okay, and this cell will be genetically similar to the parent cell. Cell will be genetically similar to the parent cell. Okay, so what is happening during mitosis? The chromosome, they replicate first, okay, and then they get equally distributed into the two daughter cells. Okay, we know at the end of mitosis, two daughter cells are formed. These two daughter cell, okay, so 2n, this was the parent cell, it will give rise to two daughter cells, 2n and 2n with same number of chromosome, okay. All right. So this is why this happens, equational division. This happens because the chromosomes first replicate, and so chromosome is like this, okay. It has two sister chromatids. These two are known as sister chromatids. So during mitosis, separation of sister chromatid takes place. Okay, there is separation of sister chromatids. So that's why the daughter cells which are produced are genetically similar and have same number of chromosomes as the parent cell. And that's why mitosis is known as the equational division. So here you can see during mitosis what happens. The chromosomes are replicating and they are getting equally distributed in the two daughter cells. Mitosis leads to the even distribution of the genetic material. And the daughter cells which are produced contain the same genetic information, means same chromosome number as the parent cell, okay, with identical set of chromosomes. Okay, so this is clear. Now coming to the next question, which is, we have to name the stages here. Name the stage of the cell cycle at which these events occur. Okay, the first is at which stage the chromosome, they move to the spindle equator. Second is centromere splits and the chromosomes separate. In which stage the centromere splits and the chromatids separate. Third one is at which stage of the cell cycle pairing of homologous chromosome takes place. And the fourth one is at which stage crossing over between homologous chromosome is seen. Alright. So let's talk about the answers of this question. So first one, the chromosome move to the equator, okay, during the metaphase stage of the cell cycle. Okay, so what happens? If this is the equatorial plate, the chromosome lying on the metaphase plate. Okay, these are chromosomes with true chromatids. This is the metaphase plate or the equatorial plate. Now, second, centromere splitting and the chromatid separation is seen during the anaphase stage of the mitosis. Alright, and you can say also at the anaphase 2 of the meiosis 2. Alright, so whenever the centromere will separate and the chromatid will separate, what will happen? Then we will see equal separation of the chromosome, means the chromosome number will remain the same. Alright, so if this is the chromosome, this is the centromere, okay, which has two kinetochores where the spindle fiber attached. This centromere divides and the chromatids separate to the opposite poles. Okay, the chromatid move to the opposite poles, okay, maintaining the chromosome number. This happens during the anaphase. Now, third one is the pairing between homologous chromosomes takes place, or you can say synapses. So they are asking about the synapses here. Synapses is what? Pairing of the homologous chromosome. So this takes place during the zygotine stage of the prophase. Alright, prophase 1 you can say. This happens during the zygotine stage of the prophase 1 of meiosis 1. Alright, now the next one is at which stage the crossing over is seen between the homologous chromosome. So the answer will be the Pachytene stage again of prophase 1 of meiosis 1. Alright. Now coming to the next question. Now here we have to describe these following terms. First one is the synapses, second is the bivalent, and third one is the chiasmata. And we have to explain them using the diagram. Okay, so first is synapses. Synapses is nothing but the pairing of the homologous chromosome that is seen during the zygotine stage of prophase 1. Alright, so in this figure you can see this is, okay, synapses is happening here. This is the pairing of two homologous chromosomes. This is chromosome number 1. This is the, okay, and this one is the second chromosome. So homologous chromosome pairing is known as synapses. Alright, so second one is the bivalent. Okay, due to synapses, a bivalent is formed. Okay, so this is the bivalent. Means pair of two homologous chromosome. Bivalent is also known as a tetrad. Why tetrad? Because it contains 4 chromatids. Okay. Now third one is the chiasmata. Chiasmata is nothing but the point where the homologous chromosomes are attached. Okay. And these are X-shaped structures. You know what happens during Pachytene? Crossing over is taking place. During Pachytene, And what happens? The homologous chromosome starts separating during the next stage, which is the diplotin. But what happens? They remain attached at the point of crossing over. Alright? And form an X-shaped structure. So this is a point of crossing over. Alright? And it is a X-shaped structure, and this is known as chiasma or chiasmata. This is seen during the diplotin stage of prophase 1. Okay. Now coming to the next question. How is cytokinesis in plant differ from the cytokinesis in the animal cell? So we already know that the cytokinesis or the mechanism of cytokinesis differs in plant and the animal cell. Now let's talk about the cytokinesis in animal cell first. So in animal cell, what happens? There is appearance of a furrow in the plasma membrane first. Okay, whereas in plant cell, there is appearance of cell plate in the center of the plant cell. Okay. So what happens during the during the cytokinesis in animal cell? If this is the cell, there is an appearance of furrow here in the plasma membrane. Okay. And this furrow deepens or it moves inwards. This furrow moves inwards, dividing the cell into two parts. Alright. So in next stage, this furrow will deepen. Okay. It will move inwards from here like this and like this, resulting in the formation of two cells in the end. This is known as the furrow method or the cleavage furrow method. Whereas in plant cell, a cell plate appears in the center first. Okay. If this was the plant cell plate appears in the center. Okay. This is the cell plate. Okay. And what happens? This cell plate then grows outwards. Okay. Towards the outer side. In animal cell, the furrow was growing inwards, whereas the plant cell, it is growing outwards. And finally, it forms two cells in the end. Okay, so this cell plate moves outwards and reaches the lateral walls, okay, and forms the two cells in the end. So this was about the mechanism of cytokinesis in animal cell and plant cell. Here you can say animal cell cleavage furrow method, or you can say furrowing method. Here you can say cell plate method or cell plate mechanism. Now let's talk about the next question, which is we have to find example where the four daughter cells from meiosis are first one, they are equal in size. Okay. And second example we have to give where the cells are found to be unequal in size. Alright. So the answer for the first question where the four daughter cells are equal in size, that will be during spermatogenesis. So what is spermatogenesis? Spermatogenesis is formation of sperms. Alright. So during spermatogenesis, okay, meiosis takes place. We know during gametogenesis or formation of gametes. And during meiosis, what happens? One cell first will give rise to two cells. Okay, if this was 2n, after meiosis 1, two daughter cells are formed, which are haploid, chromosome number are reduced, and this cell will further give rise to two daughter cells again, which are again haploid, because meiosis 2 is equational division. So 4 cells are produced, and during spermatogenesis, these are equal in size. Okay. So 4 equal sperm cells are produced. Okay, due to meiosis. Now an example, okay, where unequal size cells are formed will be during the oogenesis, means formation of ovum. Okay, so oogenesis result in the formation of 4 daughter cells which are unequal in size. Alright, so what happens during oogenesis? One big ovum means the egg cell, okay, whereas 3 small polar bodies are formed. Okay, so this was some of the examples where unequal and equal size cells are formed during meiosis. Now coming to the next question, here we have to differentiate between the anaphase of mitosis from the anaphase 1 of meiosis. Alright, so let's talk about the anaphase of mitosis first. So during anaphase of mitosis, there is a separation of sister chromatid. Okay, that's why it is known as equational division. Okay, separation of sister chromatid takes place, the centromere splits, okay, and 2 genetically equal cells are formed which have the same chromosome number as the parent cell. Alright, whereas during the anaphase 1 of meiosis 1, okay, separation of homologous chromosome is taking place. Alright, that's why it is known as the reductional division. It is known as the reductional division. Alright, so here chromosome, if the organism contains 46 chromosome, 23 will go on in one cell, 23 will go in the other cell. So this was diploid. So haploid condition is attained after anaphase 1 of meiosis 1. Alright, whereas in anaphase of mitosis, if the cell was diploid, it will still remain diploid due to the separation of sister chromatids. Now, coming to the next question. Now here we have to list the main differences between the mitosis and meiosis. Okay. So now let's talk about the differences between mitosis and meiosis. Mitosis takes place in the somatic cells, also known as the vegetative cells. Okay. Or you can also say these somatic cells and vegetative cells are usually diploid. Okay. They are 2n, or you can say they are usually diploid. Okay. Whereas meiosis is known as the reductional division, and it takes place in the germinal cells. Okay. Or you can say in the reproductive cells. Okay. Or you can say meiosis takes place during gametogenesis, formation of gametes. It takes place during gametogenesis. Now, mitosis is known as the equational division because there is a separation of sister chromatids during mitosis, whereas meiosis is known as reductional division because there is separation of the homologous chromosome during meiosis. Now, mitosis, the prophase of mitosis is not that much complex. It is okay, and it is not further divided into any substages. Alright, whereas the prophase 1 of the meiosis 1 is further divided into 5 substages. It is longer and complex than the prophase of mitosis, and it is further subdivided into leptotene, zygotine, pectine, diplotin, and diakinesis. Now, some other difference between mitosis and meiosis are, first of all, no recombination, no crossing over, and no chiasmata formation is seen during mitosis. Means, alright, if there is no recombination, no crossing over, what happens? There is no recombinants formed. Okay, no recombinants are formed because there is no exchange of genetic material. Exchange of genetic material which happens during the crossing over. Alright, so ultimately means there is no variation. Alright, whereas in meiosis, recombination is seen, crossing over seen, and chiasmata formation also occurs during meiosis. And this will lead to variation, genetic variation, which finally leads to the evolution of a new species. This variation will accumulate and over the time it will lead to evolution of a species. Alright, in mitosis what happens? It results in the formation of two genetically equal daughter cells, okay, with same number of chromosomes, whereas in meiosis, four unique, you can say, daughter cells are formed which are haploid. Okay, so these four unique cells are haploid, means contains one set of chromosome. Alright, so this was some of the main difference between mitosis and meiosis. Now let's talk about the significance of meiosis. So next question is, what is the significance of meiosis? Why meiosis is important? Alright, so first will be meiosis leads to the conservation of the chromosome number of any species which is sexually reproducing. Okay, so meiosis helps to maintain the chromosome number in any species which is sexually reproducing. Okay, so meiosis we know takes place during gametogenesis, means formation of gametes, and the two gametes, suppose gametes are haploid, so these two gametes are produced due to meiosis, and after fertilization what happens? Diploid condition is maintained, okay, and these gametes were produced due to meiosis. We can write gametogenesis. In which meiosis takes place. Alright, initially the cell was diploid. Okay, after meiosis, haploid cells are formed, and after fertilization, the diploid condition is maintained. So this is how meiosis leads to the conservation of the chromosome number of a species. Second one is that meiosis helps in the generation of the haploid spores, okay, and gametes. Okay, gametes are also haploid. Again, most important point is here is that meiosis is the source of variation. Okay, it is the source of genetic variation, and variation will accumulate over time and will lead to evolution of a new species. And why it is a source of variation? Because of the crossing over that is taking place during the meiosis. Alright, and in meiosis what happens? Okay, independent assortment of chromosome is taking place. Independent assortment of both paternal and maternal chromosome is taking place, which leads to the formation of new combinations. Okay, new combinations are produced, Okay, or we can say variations are produced. Again, due to the independent assortment of the chromosome. The chromosomes separate independently of each other, or you can say chromosome which specify different traits, okay, they assort independently of each other and form new combination in the daughter cells. So this was about the significance of meiosis. Now, the next question is you have to discuss with your teacher. First one is the haploid insects and lower plants where cell division occurs. Okay, second one is we have to mention that the cell division occurs where and the second one is we have to mention some haploid cells in higher plants where cell division it does not occur. All right. So let's talk about the first question first. Okay. Here we have to mention the haploid insects or lower plants where cell division occurs. So first one, let's talk about the haplontic life cycle first. So in haplontic life cycle, what happens after the fertilization? Okay, if this was two gametes, after fertilization, a zygote is produced. Okay, and what happens? Meiosis takes place in this zygote. Okay, meiosis occurs in this zygote, leading to the formation of haploid organism. So meiosis will reduce the chromosome number to half. Okay, and then haploid organism is produced. All right. So gametes are produced via mitosis in some of the organism. Okay, and some of the examples are, first is the algae. Okay, algae is a kind of a plant. All right, so in algae, mitosis leads to the formation of haploid organism, and in some insects as well, in some insects such as honeybees, wasps, and ants, okay, mitosis also leads to the formation of sperms here. Okay, so what is happening? Let's take an example of honeybees. So honeybees, what happens? The male drones, okay, they are haploid because they are produced by the process of parthenogenesis. What is parthenogenesis? Parthenogenesis is formation of organism without the fertilization. So they are produced without fertilization. Okay, so the egg is not fertilized and it directly develops into male drones. Egg is haploid. Okay, it develops into male drones because of parthenogenesis, and these male drones, with the help of mitosis, produce sperms. Okay, usually meiosis takes place during the formation of sperms, but here mitosis is taking place in male drones and leading to the formation of haploid sperms. All right. So now let's talk about the second part of the question. Okay. So in some cells of the higher plant, cell division does not occur, okay, leading to the formation of polyploid cell. Okay. And what are polyploid cell? Polyploid cells are cells which contain multiple set of chromosome. Okay, if the cell initially was diploid, means it contains two sets of chromosome number. So sometimes cell division will not take place, okay, or you can say there is no cytokinesis, there is no cell division, okay, and all the chromosome they go to the one cell, okay, and an polyploid cell is produced. okay, which can contain, suppose, 4n here. Okay, double the number of chromosome sets. Alright, so polyploid cell contain multiple sets of chromosome. Okay, or you can say they contain chromosome which are typically more than the typical diploid number. So these polyploid cells are produced in some of the higher plants, okay, due to the lack of the cell division. Okay. Now let's talk about the next question, which is, can there be mitosis without the DNA replication or the DNA synthesis in the S phase? Let me again mention about the interphase, phases of interphase, G1 phase, S phase and G2 phase. DNA synthesis takes place here during the S phase. Alright. So here they are asking whether the mitosis can occur without the DNA replication. The answer will be that the mitotic cell division cannot take place without the DNA synthesis during the S phase. Okay. So what happens? The DNA duplication is important because it maintains the chromosome number in the daughter cells of a sexually reproducing organism. Alright. So what happens during mitosis? 2n is the parent cell. Okay. Again, daughter cells are produced, 2 daughter cells which contain the same number of chromosomes. Alright, this happens because of the DNA duplication that is taking place during the S phase. Okay, and the chromatids separate during the mitosis, thus maintaining the chromosome number. And that's why mitosis is also known as the equational division. If DNA synthesis doesn't take place, then what will happen? Then chromosome will separate instead of chromatids. Okay, then the chromosome will separate instead of chromatid and it will reduce the chromosome number to half. Okay, so DNA duplication is very important. Without that, mitosis cannot take place. Now, another question is, can there be DNA replication without the cell division? Alright. So this is the opposite of the previous question. So the answer will be yes. DNA replication can take place without the cell division, and this occurs in cell known as the polytene chromosomes. Okay, this takes place in some of the cells and the process is known as polyteny. Okay, so what happens during the formation of this polytene chromosome? There is the failure of separation of the replicated strands of DNA, or you can say there is the failure of the separation of the chromatids. And this happens repeatedly, again and again. The chromatids don't separate, okay, and they give rise to polytene chromosome. Okay, so example of DNA replication without cell division will be, you can say this can be seen in polytene chromosome, where the chromatids don't separate, okay, and this happens again and again, and this leads to the formation of a cable-like structure. Okay, this leads to the formation of a cable-like structure. So chromosome appear like this. Okay, it appears like this. These polytene chromosome appear like this. This is the main axis of the chromosome, and these are the loops of DNA. Okay, that is dividing again and again, but they are not separating. So these polytene chromosomes are observed in the order Diptera, which belongs to the class Insecta of the phylum Arthropoda. For example, the Drosophila salivary glands. Okay, Drosophila Okay, the model organism in the animal kingdom. Okay, the salivary glands of Drosophila. Okay, this is the example of the cells where DNA replication takes place without cell division. Now, coming to the last question of your NCERT book, which is, here we have to analyze the events, okay, during every stage of the cell cycle. Okay, and we have to notice how the following parameters change during the cell division. So first one is the number of the chromosome, whether they are changing or not. Second is the amount of DNA content that is changing per cell. Okay, so first let's talk about the chromosome number. Okay, how the chromosome number can change, okay, during mitosis or meiosis. So now let's talk about the first part of the question, which is number of chromosome per cell, how it changes. So now we know that the chromosome count or the chromosome number of the diploid cell is 2n. Okay. So what happens during the anaphase 1 of meiosis? The homologous chromosomes separate. Okay. The homologous chromosomes separate during the anaphase 1 of meiosis, reducing the chromosome number to half in the daughter cells which are produced. Okay, initially the cell was diploid. After anaphase, the cells will have haploid number of the chromosome. Okay, so here you can see the chromosome number have changed. Alright, and what happens during meiosis 2? Okay, meiosis 2 will take place after this, but the chromosome number will be same now. Here there is separation of the chromatids. That's why the chromosome number remains the same. Here there is separation of the chromosomes. Okay. So we already talked about the anaphase 1. Let's talk about the meiosis 2. Meiosis 2 maintains the same chromosome count as the sister chromatids separate here. Therefore, at the end of meiosis, daughter cell contain half the number of chromosome. Okay, so at the end of meiosis, okay, 4 cells are formed with half the number of chromosome of the parent cell. Now let's talk about the change in the DNA content per cell. Alright, so DNA synthesis which takes place during the S phase. DNA synthesis takes place during the S phase. So what happens? In G1 phase, if the cell was diploid, it contains 2C amount of DNA. Okay. In S phase, what will happen? The chromosome number will remain the same. But what will happen? The DNA content will double and it will become 4C. Alright, so in the G2 phase, we can say the cell have double the amount of DNA, but the chromosome number remains the same. Okay. So consider the initial DNA content of the cell, okay, as 2C. Okay, 2C. Okay, during the S phase, what happens? DNA synthesis takes place. Okay, doubling the DNA content to 4C. Okay. And what happens at the end of meiosis 1? The DNA content in the cell is once more reduced to half. Okay. So we had interphase which was divided into G1, S phase and G2 phase. After interphase, we have meiosis 1. Okay. Here the DNA content was 4C. But after meiosis 1, what happens? Homologous chromosomes separate and it will lead to the reduction in the DNA content because 2 cells are formed. Okay. So they will have half the number of DNA content, 2C and 2C. Now what will happen? This will again undergo meiosis 2. And what will happen? 4 cells will be produced. Okay. And each of these cells will now contain C amount of DNA. Okay. They are haploid. Okay. These are also haploid. This was diploid. Okay. So here you can see after the meiosis 1, the DNA content of the cell will reduce to half. Okay. And 2C condition will be restored again. And then after meiosis 2, the DNA content is further halved. Okay. And what happens? Resulting in the formation of cells having DNA content of C. So this is how the DNA content of the cell changes during the cell cycle or during the meiosis. So this was the end of today's lecture. We have completed all the 16 questions mentioned in the NCERT book. And we will meet again next time. Thank you for listening. Thank you.